Good day, my name is Sherilyn M. Otos, and for today's video, I am going to be discussing all about photosynthesis. Photosynthesis. Photosynthesis is the process by which plants, some bacteria, and some protistants use the energy from sunlight to produce glucose from carbon dioxide and water. This glucose can converted into pyruvate, which releases adenosine triphosphate or ATP by cellular respiration. Oxygen is also formed. Photosynthesis. Photosynthesis may be summarized by the word equation that is shown. The conversion of usable sunlight energy into chemical energy is associated with the action of the green pigment chlorophyll. Chlorophyll is a complex molecule, and several modifications of chlorophyll occurs among plants and other photosynthetic organisms. All photosynthetic organisms have chlorophyll. Accessory pigments absorb energy that chlorophyll A does not absorb. Accessory pigments include chlorophyll B, also C, D, and E in algae and protistants. Xanthophylls and carotenoids such as beta-carotene. Chlorophyll A absorbs its energy from the violet blue and reddish orange-red wavelengths, and little from the intermediate green, yellow, orange wavelengths. All chlorophylls have a lipid-soluble hydrocarbon tail or C2OH39, a flat hydrophilic head with a magnesium ion at its center, and it has different chlorophyll have different side groups on the head. The tail and head are linked by an ester bond. Leaves and leaf structure. Plants are the only photosynthetic organism to have leaves, and not all plants have leaves. A leaf may be viewed as a solar collector crammed full of photosynthetic cells. The raw materials of photosynthesis, water and carbon dioxide, and it enters the cell of the leaf, and the products of photosynthesis, sugar and oxygen, leave the leaf. Water enters the root and is transported up to the leaves through specialized plant cells known as xylem vessels. Land plants must guard against drying out and so have evolved specialized structure known as stomata to allow gas to enter the leaf. Carbon dioxide cannot pass through the protective waxy layer covering, which is the cuticle, but it can enter the leaf through the stoma, the singular of stomata, flanked by two guard cells. Likewise, oxygen produced during photosynthesis can only pass out of the leaf through the open stomata. Unfortunately for the plant, while these gases are moving between the inside and outside of the leaf, a great deal of water is also lost. Cotton trees, for example, will lose 100 gallons of water per hour during hot desert days. The structure of the chloroplast and the photosynthetic membranes. The thylakoid is the structural unit of photosynthesis. Both photosynthetic prokaryotes and eukaryotes have these flattened sacs or vesicles containing photosynthetic chemicals. Only eukaryotes have chloroplast with a surrounding membrane. So this is the structure of a chloroplast. Thylakoids are stacked like pancakes in stacks known as grana. The area between grana is referred to as stroma. While the mitochondrion has 2 membrane stems, the chloroplast has 3, and it forms 3 compartments. Stage of photosynthesis. When chlorophyll A absorbs light energy, an electron gains energy and is excited. The excited electron is transferred to another molecule called a primary electron acceptor. The chlorophyll molecule is oxidized or loss of electron and has a positive charge. Photoactivation of chlorophyll results in the splitting of water molecules and the transfer of energy to ATP and reduce nicotinamide adenine dinucleotide phosphate or NADP. The chemical reaction involved includes condensation reaction. It is responsible for water molecules spitting out, including phosphorylation, the addition of phosphate group to an organic compound. The next one is oxidation reduction or redox reaction involving electron transfer. Photosynthesis is a two-stage process. The light-dependent reaction, a light-dependent series of reactions which occur in the grana and require the direct energy of light to make energy carrier molecules that are used in the second process. Light energy is trapped by chlorophyll to make ATP or photophosphorylation. At the same time, water is split into oxygen, hydroions, and free electrons. The electron then reacts with the carrier molecule nicotinamide adenine dinucleotide phosphate or NADP, changing it from its oxidized state to its reduced state. The light-dependent reaction, a light-independent series of reactions which occur in the stroma of the chloroplast when the products of the light reaction, ATP and NADPH, are used to make carbohydrates from carbon dioxide reduction. Initially, glyceraldehyde 3-phosphate, a 3-carbon atom molecule, is formed. Light-dependent reaction. When light energy is absorbed by a chlorophyll molecule, its electrons gain energy and move to higher energy levels in the molecule, or photoexcitation. Sufficient energy ionizes the molecule with the electron being freed, leaving a positively charged chlorophyll ion. This is called photoionization. So, in pool chloroplast, each chlorophyll molecule is associated with an electron acceptor and an electron donor. These 3 molecules make up the core of photosystem, and 2 electrons from photoionized chlorophyll molecule are transferred to the electron acceptor. The positively charged chlorophyll ion then takes a pair of electrons from neighboring electron donors such as water. So, this is a condensation reaction that has led to phosphorylation. An electron transfer system, or a series of chemical reactions, carries the 2 electrons to and fro from across the thylakoid membrane. The energy to drive these processes comes from 2 photosystems. The first one is the photosystem 2, PS2 or P680, and the photosystem 1, PS1, P700. It may seem confusing, but PS2 occurs before PS1. It is because It was the second to be discovered and hence named second. The energy changes accompanying the two sets of changes that make a Z shape when drawn out. This is why the electron transfer process is sometimes called the Z scheme. Key to the scheme is that sufficient energy is released during electron transfer to enable ATP to be made and from ADP and phosphate. Non-cyclic phosphorylation, the Z scheme. Both adenosine triphosphate or ATP and NADPH are produced in the first photosystem or photosystem 2, PS2. Photoionization of chlorophyll transfer excited electrons to an electron acceptor. Photolysis of water, an electron donor, produces oxygen molecules, hydrogen ions, and electrons, and the latter are transferred to the positively charged chlorophyll. The electron then acceptor passes the electron to the electron transport chain. The final acceptor is photosystem PS1. Further absorb light energy increases the energy of the electron sufficient for the reduction of NADP plus to NADPH. So in this picture, the oxidized form of nicotinamide adenine dinucleotide phosphate. And this one is the reduced form of nicotinamide adenine dinucleotide phosphate or NADPH. So, chemiomeiosis and ATP synthesis. The components of non-cyclic phosphorylation are found in the thylakoid membrane of the chloroplast. Electrons passing through the transport chain provide energy to pump H+ ion from the stroma across the thylakoid membrane into the thylakoid compartment. H+ ions are more concentrated in the thylakoid compartment than in the stroma. We say there is an electrochemical gradient. H+ ion diffuse from the high to the low regions of concentration. This drives the production of ATP. Chemiomeiosis as it operates in phosphorylation within a chloroplast. Cyclic phosphorylation, the net effect of a non-cyclic phosphorylation is to pass electrons from water to NADP. So, the energy released and it is enabled to produce ATP, but much more ATP is needed to drive the light-independent reactions. So the cycle is completed by electrons being transported back to PS1 by the electron transport system. The light-independent reaction. So this is also called dark reaction. The carbon dioxide from the atmosphere is captured and modified by the addition of hydrogen to form carbohydrates. The incorporation of carbon dioxide into organic compound is known as carbon fixation. So living system cannot directly utilize light energy but can through a complicated series of reactions, and it converts into CC bond energy that can be released by glycolysis and other metabolic processes. So carbon dioxide combines 5-carbon sugar and ribose 1,5-biphosphate or RUBP. This one is the glycerate triphosphate or GT molecules. It is phosphorylated by ATP into glycerate diphosphate molecules. So this is a reduced by NADPH to 2 molecules of glyceraldehyde triphosphate. Of each pair of GALP molecules produced, one molecule is the initial end of the photosynthesis. It is quickly converted to glucose and other carbohydrates, lipids, or amino acids. So the first step in the Calvin cycle is the product of the Calvin cycle is the phosphoglycerate, and the energy from ATP and the NADPH energy carriers generated by the photosystem is used to phosphorylate the PGA. And eventually, there are 12 molecules of glycealdehyde phosphate, also known as phosphoglycerolide glycealdehyde or PGAL, two of which are removed from the cycle to make glucose. So the factors affecting the rate of photosynthesis is the following: the carbon dioxide concentration and temperature. It is known as the limiting factors. An increase in the carbon dioxide concentration increases the rate of which carbon is incorporated into carbohydrate in the light-independent reaction, and so the rate of the photosynthesis generally increases until limited by another factor. So photosynthesis is dependent on temperature, and it is a reaction catalyzed by enzymes. And as the enzymes approach their optimum temperature, the overall rate increases. Above the optimum temperature, the rate begins to decrease until it stops. So that is the end of the video. Thank you for watching and have a great day.